expression does not exist when it comes to our cells. Instead, the making of proteins from the level of taking their recipe all the way to a final protein is tightly regulated, and even once you make the protein, it's regulated after that. So there are lots of different levels of genetic regulation. And so when we talk about like gene expression, we're often talking about things at different levels depending on what types of experiments we're doing. So I want to tell you more about these types of experiments we can do to get kind of at the question of how much is a gene product getting made, and we can look at whether like it's starting out, getting a running start, or if it's making it all the way to the finish line, and then all the way in between. There are different ways that we can look at the various steps in the process, and this gives us information not just about like how much of a product is getting made, but where there might be holdups, and that sort of thing, where regulation might be going on, and various ways that we can gain more insight into these various processes going on inside of our cells.
So for the sake of simplicity, and a lot of these techniques that I'm talking about are going to be protein specific, but note that when we talk about expression, we can also have expression of like functional RNAs and things that genes, recipes. So genes are these genetic recipes for making a like functional product. So often these are proteins, but they're not always. And in some cases, they're things like functional RNAs, which have important roles, such as like microRNAs and stuff. These have important roles, but they function at the level of the RNA. Whereas for proteins, RNA is just an intermediary between the like original recipe for the protein, which is in DNA, it's the gene, and the final product, the protein. So in our cells, we have basically these instructions for all of these different proteins are housed in the form of DNA, and in this long coiled up DNA called chromosomes, and the various stretches of, so a protein is going to have its instructions in the form of like a stretch of that DNA. So kind of like a chapter in a cookbook. And this cookbook is very, 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 very precious because it has all of our recipes for making proteins and functional RNAs. So we don't want to touch that original recipe a lot. So we keep it safe in this membrane-bound room called the nucleus. And then in order to make a protein from it, what happens is that we make messenger RNA copies of it. So this RNA polymerase enzyme, so an enzyme is a reaction speeder upper, so RNA polymerase is going to make RNA copies of the gene, and they're going to take it out of this nucleus. It's well, not they, but it'll get taken out of this nucleus into the cytoplasm, which is like the general cellular interior. And here there's going to be protein making complexes called ribosomes that are going to, in the process of translation, go along that messenger RNA and turn it into a protein. So there's actually some processing steps in between going from a gene to a mature messenger RNA. So you go to a pre-messenger RNA, and it has these regulatory regions called introns, which are going to get spliced out, and then this gets a cap and a tail and stuff. So there's a little processing, but the basic idea is that you go gene, transcription, messenger RNA, translation, protein. And then this protein, that's not necessarily the end of the line. So this is can get modified and degraded after this translation. So we call these like post-translational modifications. So the transcription is happening in the nucleus, at least in eukaryotes, so things like us, and animals and plants and stuff, but like not bacteria and archaea and stuff. But so this transcription is happening in the nucleus, and then the rest, the translation is going to be happening in the cytoplasm. Or in certain cases, it happens inside of other like membranous compartments inside of the cell for things that need extra help. So there's some like translation going on in like the endoplasmic reticulum, so the ER and that sort of thing, for things that often need modifications and things that get shipped out of the cell and that sort of thing. Just note that in bacteria, because they don't have that nucleus where they're holding their DNA, they're actually, everything's happening kind of at the same time out in the same place in the cytoplasm. So they have transcription and translation coupled so that as they're making the messenger RNA, it's already being translated. You can imagine that the more copies of a messenger RNA you have, the more translation that you can have, and therefore the more protein that you can have. So one way that cells can regulate the levels of the final protein that they want to make is by regulating transcription to make more messenger RNA copies. They can also regulate translation, so regulates how well those messenger RNA copies are used, and then they can actually, once the protein is made, they can then regulate how long the protein stays around and various things, often by making modifications onto them, like ubiquitin if they want to tag them for destruction. So there are different levels of regulating what quote unquote expression. So often when people are talking about like measuring gene expression, they're talking about transcription. So at this, I guess at the simplest level, this would be the most expression-ish, because say like if you have like an RNA, if you have like a functional RNA, it's not even going to go through translation. It might have to go through some processing steps, but different steps. But what we often care about more is how much of the final product is actually made and how much is it sticking around. So we can measure various aspects to measure like the intermediates as well as the final product. So we'll go through more of these techniques in detail, but you can measure transcription. So basically with Pol-2 ChIP-seq, what you can do is basically just see what the RNA polymerase is bound to. You can use techniques like RT-qPCR or Northern blot, microarray, RNA-seq to measure the amounts of the messenger RNAs. So this is going to take into account both transcription and decay. Then you can measure translation with various things like polysome profiling or riboseq. You can measure the actual levels of the protein with things like a Western blot, and that'll get into translation and degradation. And so you have all of these various processes contributing, and if you look at it, you can use different techniques to get looks at different steps in the pathway. And so when we talk about gene expression, we need to kind of be aware that there are these different steps, and we want to make sure we know what level people are looking at. So often there's a good correlation between the levels of the messenger RNAs and the levels of protein, the final protein, but this isn't always true. And sometimes the most interesting, or what I think are kind of really interesting proteins, there is not the direct relationship because they're under tight control at various levels. So let's talk a little bit more about these various levels of control, and then we will go into how we can measure them with these techniques. So we'll start at the beginning with transcriptional regulation. So this is going to involve how many messenger RNA copies do you make? And so remember that this is going to, transcription is where the RNA polymerase is going to bind in front of the gene. It's going to transcribe a messenger RNA copy that's going to get used to make a protein. But right now we're looking at this transcription. So we need this RNA polymerase needs to bind to this region ahead of the gene called the promoter. And then it needs to transcribe the gene. So this promoter is often going to be hidden until the gene, it's time to express the gene. So inside of our cells, we have a ton of DNA. And so we have to coil it up really tightly, or else it wouldn't even fit in the side of our cells. So how this is coiled up is it's done with the help of these histone proteins. And so these histone proteins, these are proteins that act a bit kind of like hair rollers for DNA. So this saves space and it hides the DNA that it's wrapped around. So there's these octamers. So there's like 8 little proteins, and the DNA wraps around them to form these like nucleosomes. So if you imagine that the promoter region is somewhere in here, but it's all hidden. So to access the DNA, there are complexes that can come and they can kind of like slide the histones out of the way to make that region accessible, or they can remove the histones entirely. And this is going to help regulate access to transcription factors, so things that are going to kind of like help RNA polymerase bind, or maybe even prevent it from binding, and polymerases. So, so how these histones often get like modified epigenetically. We call this like epigenetic modifications, but they're like post-translational modifications to the lysine tails of these proteins. So the amino acids of the protein letter lysine, these tails, the tails of these proteins are really rich in them. And so they can get modified with things like methylation or acetylation that can change how tightly the DNA is wrapped around it. And they can also provide binding sites for various chromatin remodelers that can come and kind of push things away and make changes, and therefore help regulate the level of expression at the level of transcription. So chromatin, we call it like chromatin, where you have kind of chromatin's just like the chromosomes, especially when they're like wound up and stuff. And so chromatin remodelers are going to kind of be those complexes that are going to help move the nucleosomes and that sort of thing. And so by doing this, they can regulate access to the promoter regions and to the genes. And often these transcription factors are these proteins that are going to bind to promoter sequences in front of the genes, and they help recruit RNA polymerase. And you can also have like things like enhancer regions and various things where regions of the DNA are kind of going to help fold things up and bring regions together. And there's a lot of really cool stuff going on when it comes to regulating transcription. And it's a very active area of research, especially because transcription factors are sometimes, they can be targets for various therapeutics, if there is sort of misregulation or making too much of a various protein in a disease or that sort of thing. So there's a lot of work being done to kind of take advantage of some of the specificity of transcription factors. So transcription factors aren't just generic, different proteins. Transcription factors often regulate similar like sorts of genes in terms that they can act on things that you want to be expressed at the same time, but then they can also, you can imagine that if you have transcription factors that are going to be regulating some process that is really important, that like a cancer cell would hijack, then you can potentially target the transcription factor in order to try to dampen down the expression. So various different things, so just something to be aware of for in the future, that this is an area of active research that a lot of people are looking into. And we could have more and more drugs that actually try to target these transcription factors. So now we've talked about transcription, and later we'll talk about how we can measure that. But once we have transcription, so when we're just going to skip over this processing phase, so we're just talking about having mature messenger RNA. This messenger RNA can now get regulated post-transcriptionally. So after it's been made, it's still like, is it sticking around? So this is going to depend on things like microRNA, which is my favorite topic because this is what I study. So microRNA is part of this RNA interference or RNAi pathway. So I'm not going to, I know this looks like a ton of information, and I'm not going to go into all of this because I have oodles of posts on this, and I'm not going to bore you with it again. But the basic idea is that you have this Argonaute protein, and it's going to be directed to specific messenger RNAs based on their sequence. So this Argonaute protein binds to a small RNA, like a microRNA, and then uses that as a guide to find and bind messenger RNA targets that have complementarity to this guide. And then it can, either alone or with the help of cofactors, trigger their degradation and repress translation. So it's acting at the level of this messenger RNA. And so in our cells, this is done by with microRNAs. So we have genes for these microRNAs that make those small RNAs that bind to the argonaute protein. So this is actually regulating the messenger RNA in a region that isn't actually protein coding typically. So this is happening in the 3' untranslated region. So in addition to the protein coding regions of the messenger RNA, so the regions that actually have instructions for making proteins, there's a little bit of regulatory information. So I talked about how we took out the introns, so those regions in between the genes, but they leave in some of these 2 regions. So at the beginning, like before the protein instructions and after the protein instructions. So here you have the 5' untranslated region, or the 5' UTR, and here you have the 3' untranslated region, the 3' UTR. And 5' and 3' just refer to the different ends. So you have this 3' UTR, this untranslated region after the protein coding region. And this is typically where you have these microRNA binding sites. So these like target sites. So different proteins will have messenger RNAs with different combinations of sites and different sites for different microRNAs, and they can have sites for multiple microRNAs, and the same microRNA can have a site in multiple messenger RNAs. So this allows you to have like combinatorial regulation. So in our cells, we're using these microRNAs, but we can also introduce like externally small interfering RNAs or siRNAs that we can then get AGO to use to target specific genes or specific messenger RNAs, and this can be used to like knock down expression. So it's an important experimental technique that's really cool too. So yeah, so that was my plug for RNAi, and much more on it in other posts. So the levels of your messenger RNA are going to depend on how much you're making it and how much you're degrading it. And so even if a messenger RNA isn't being degraded by this RNAi, they still have like ways to regulate without it, some involving things going on often with the tail length and that sort of thing, although the exact relationships aren't exactly clear. But there is regulation, and there are other proteins that can target messenger RNAs for degradation, and there are also a lot of pathways that can target faulty messenger RNAs for degradation as well. But basically your cells have ways to regulate how long these messenger RNAs are sticking around, so that they don't just, because you can imagine if you just kept making things at various times, so you're controlling when you want to make the messenger RNA, but then you need to be able to control when you want to get rid of it. So even if you stop making it, if you were just to keep building it up, that wouldn't be good, because you'd be making stuff that you wanted like yesterday. Okay, so now though, we need to actually make protein from this. So now we're talking about translational regulation. So the amount of protein that we make is going to depend on the amount of messenger RNAs that we make. But it can also depend on other things, features of the sequence that can help promote translation or make it harder. So in translation, the ribosome is going to be moving along the messenger RNA, and it's using these like 3-letter RNA reading these 3-letter RNA like words or codons. And so a tRNA, a transfer RNA with a matching, with an anticodon, is going to see a codon in this active site of the ribosome, and it's going to come and bring in the corresponding amino acid. But there's redundancy in this code, so that multiple codons can spell the same amino acid. And cells it's not always like, can be differerences in the usage of various codons. So even though these codons all spell the same thing, because they correspond to transcription, or because they correspond to tRNAs, then they have different tRNAs often, then cells might stock up on different tRNAs some tRNAs more than others. This can give you like codon usage bias. So if you were to look at the usage of the codons that all spell the same letter, that all carry the same amino acid, recognize the same, but they recognize different codons, there won't be like an even mix of them. And so cells can they're in one way that you can have regulation is by regulating which amino acids are, which codons are used, this can actually influence the speed of translation. That's not all though. There's also really important things about sequence features. So if there's weird structures in the messenger RNA, like secondary structure, so the RNA can fold up and make bonds to itself. And this might make it harder to access for various, for RNA polymerase or that sort of thing. There is also, yeah, so basically there could be the other things that influence the translation that aren't just about having like too many rare codons or that sort of thing. And so I talked about this in my post the other day on codon optimization, because when we want to get cells to make a protein recombinantly, so basically we stick in the instructions for making a protein and ask the cells to make it for us. So often we're sticking these recipes for like a human protein into a bacterial cell, and because they had different codon usages and because that sort of thing, we want to optimize our codons that we're using in the instructions that we give them. But we also have to optimize to make sure we're not adding extra things. So basically, look at the post on codon optimization if you want to know more. When it comes to like evolution and mutation rates and that sort of thing, there's a lot of focus on the amount of substitutions that are non-synonymous, so making protein level changes. And there's often this seems to be this thinking that only those changes are going to really matter. But, and same for like when you're looking at disease risks or things, are there mutations that are actually changing the protein letter? But I wonder a lot about whether changes might be like affecting translation and that sort of thing, that there could be changes that we really don't appreciate as much. And they probably have like a lot less of an effect, but I'm just kind of curious about it. Yeah, so I'm hoping in the future, maybe there'll be more research done in this sort of thing. I'm guessing it's a lot harder, but that's just something that I think about sometimes as I learn more and more about translation. But back to our story. So we've done the translation, we've made the protein, and now what happens? So this protein, it goes about its business, and it can get modified. So we can have post-translational modifications. There's certain amino acids and certain protein letters that can get changed in various ways. They can be phosphorylated. They can be glycosylated, so have sugar chains added. So there are various modifications that can kind of make a protein, just help regulate the protein's activity. But there are also modifications that can help make the protein like target it for degradation. And so there's this ubiquitin proteasome system, where basically proteins, proteins that there could be a problem with the protein, maybe it's misfolded or it's incomplete, but it also might just be that you don't need it anymore. And so you can tag these proteins for degradation by adding a little protein called ubiquitin onto the end of it. And so you can have these like chains of ubiquitin put in and that sort of thing by these ubiquitin ligases. And what this is going to do is when you add these chains of ubiquitin, then it's basically going to target it for this protein chewer complex called the proteasome. And so these ubiquitin chains are going to get put on, and then this is going to allow it to bind to this proteasome, and then the proteasome is going to chew it up, and so you get rid of the protein. And you can have ways to regulate when the protein gets ubiquitinated. So there could be other modifications or various things that are going to help or regulate the binding to these ligases and then their ultimate degradation. So you can have you can have modification all the way to the protein's end, or you can have regulation all the way to the protein's end. And so we have all of these different levels of regulation, and we can measure it in these different ways. So keep in mind this general principle that we have the gene, we have transcription, we have the mRNA, we have translation, we have the protein. And we can look at all these various steps. So let's start at the beginning. So we'll start with measuring transcription. So one of the key ways that transcription is measured is by using techniques like ChIP-seq, so like Pol2 ChIP-seq. So RNA polymerase 2, RNA Pol2 is like the protein, is the polymerase, so this enzyme that makes this DNA dependent RNA polymerase, so it makes RNA copies of DNA. So it's making those pre-messenger RNAs. And Pol2 is like the main one that makes the messenger RNAs that transcribes for proteins, transcribes the genes for proteins. So there's other polymerases that focus on things like ribosomal RNA and stuff, but Pol2 is the one that's doing this protein work. And so the basic idea with ChIP-seq, with this Pol2 ChIP-seq, is it's going to use antibodies against RNA polymerase 2. And so antibodies, we probably heard a lot about antibodies in recent years, and we often think about them in the context of viruses, but we can actually make antibodies against anything. They're just these little proteins that happen to have a sequence that the structure that is going to make them bind to something specifically. So basically these B cells are going to play around with changing this variable region of this antibodies, these little proteins. So these B cells will like randomly choose by editing their DNA, which amino acids to put on the end of these variable regions, and then the ones that happen to bind will get selected for. So we can either by so often with labs, in addition to making antibodies to like foreign things that happen to come into our body, like a virus, we can actually get animals to make antibodies against some sort of thing that we put in there. And so we call things that get bound by antibodies antigens. So you can inject an animal with something you want to act as an antigen, and then the lab animal will make antibodies. And then you can isolate those antibodies, or you can isolate the cells making the antibodies, and then make more and more of them. There are also techniques that can kind of like randomly make a bunch of antibodies. So in the future, we can do this in different ways that aren't as invasive. There are also ways to make antibodies with like yeast and that sort of thing. So there's lots of cool stuff to make antibodies that I'm not going to go into, but the basic idea is that you make an antibody that is specific for the thing that you want to bind. So a good antibody is going to bind the thing that you want and not bind other things. Why you want to have this antibody against Pol2 is that then you can use it to kind of isolate Pol2 and then see what it's bound to. So the RNA Pol2 is going to be bound to regions, hopefully, that are actively transcribing. And so if you cross-link it, or you basically freeze it onto the DNA, and then you sequence that, then you can then see where it's bound to and where it's transcribing. There are also different techniques where you can actually measure the RNA that's coming out of the polymerase. So now we can look at the levels of those messenger RNAs that are actually made. So there are different ways to do this. So with a common way, if you have a specific messenger RNA or a few of them that you were interested in, then you can use this technique called RT-qPCR to actually look at the level of that specific messenger RNA. So as we discussed earlier, the more messenger RNA copies you have, the more protein you can make, although there's not like a direct one-to-one correlation, and the correlation between the two can be different for different proteins. But so if we can measure the levels of these messenger RNAs, we get a sense of how much protein is getting made, hopefully, and it's one level of expression. And so we can use this technique called RT-qPCR in order to measure these levels. So the RT stands for reverse transcription. Sometimes it stands for real time, so you can have like RT-qPCR. This is basically for different names. But the basic idea is that you're going to reverse transcribe. So you're going to make a DNA copy of this messenger RNA. And then, so this you'll make this complementary DNA, the cDNA. And then you'll use PCR to make copies of a region of them. And so the Q in qPCR is that it's quantitative. So it's basically going to count the recipes as they're getting made, typically using like a fluorescence probes. And so you're then able to see how many copies it's making. Much more on this in my post on qPCR. But the basic idea is that it's allowing you to make lots of copies of the specific region that you want. So basically in PCR, you have to specify the region that you want copied. And so you in a sequence specific way. And so you use these primers to specify the region. And these primers are short little pieces of DNA that are going to match the region and tell the polymerase, that's the DNA polymerase this time, where to start making the copy. And so because you can use sequences that target specific messenger RNAs, you can therefore measure levels of specific messenger RNAs, but you have to do this individually. Another technique is a northern blot. So a northern blot is, again, you're looking for a specific messenger RNA. The basic idea is that you take a mix of RNAs, then you use gel electrophoresis to separate them by size. So now you have all of these RNA pieces inside of a gel, which is not a very good place to look for things, especially because they can kind of just diffuse around. So they might like wander out of the gel. Not good. So we need to transfer it to a membrane that's more durable. And so in this blot step, the RNA is going to get like wicked onto this membrane. And so the RNA is going to stick to this nylon membrane, and then you immobilize it. So you like cross-link it or with UV light or heat to get it super stuck to this membrane. And then you block it. So you kind of coat it with generic RNA. And now you can probe for RNAs of interest. So you add a labeled DNA that complements the RNA that you're looking for. And then you can see how much of the specific RNA is there. So typically these probes are like radioactive probes. So they have a radioactive phosphorus group, and this is going to allow you to visualize the products. There are also techniques like a microarray, where it's basically you have this sort of thing going on, but in lots and lots of wells of a plate, and not like radioactive. These are typically fluorescent. Then more and more these days, you see RNA-seq. This is going to measure all the messenger RNAs. And so basically, it's a high throughput type of next gen sequencing or whatever. So basically you're just measuring all of the messenger RNAs. Often it's done, you have like a poly T tail or primer or whatever for your cDNA step. So you're making copies of all of the things that have a poly A tail. So like all of the messenger RNAs. Okay, so next we can measure translation. So this can be done in different ways, including polysome profiling or riboseq. So in these techniques, you often like stop translation. So for example, by adding cyclohexamide, and you break open the cells, so you lyse them and remove the membrane gunk. And then you can so with ribosome profiling, what you're going to do is you're going to see where are the ribosomes on the messenger RNAs. And then with polysome profiling, you can see like how many ribosomes are on the messenger RNA. So with ribosome footprinting, basically you use RNases, so these ribonucleases, so these RNA chewers, to basically chew up the RNA around the ribosome. But the regions that the ribosome was bound to are going to be protected. Then you can extract the RNA that they're standing on and sequence it to see where the ribosomes are bound. And so sometimes this is called like riboseq. And so it can tell you about it can tell you cool things too, because you're seeing the regions. You can see if there are like regions that are slow, if there's like a holdup. So this is one of the ways that people know about things, like maybe they're like differences in translation rate based on codons and all of that various things. And with polysome profiling, the basic idea is that you have a lot of ribosomes translating the same messenger RNA at once. So if you have like a highly expressed thing, then it's likely to have a lot of ribosomes on it. And therefore, if you were to separate all of the messenger RNAs by their size, by like their weight or whatever, based on the ones with more ribosomes on them are going to be heavier. They're going to sink further. So you can then, by separating all of these out, and then looking to see where your specific messenger RNA was using qPCR, northern blot, et cetera, or sequencing it, you can see how many ribosomes were on each mRNA. And if there were a lot, so this is all like polysome, then you're going to see it over here. And if it's like a monosome, then it only has like a single ribosome. That's typically not as expressed. And so you can get information about that. So that's measuring translation itself. This is often done if people are interested in studying translation and various things that modify, like how sequence features and that sort of thing affect translation. But it's more common for people to measure final proteins like outside of the translation field. So if you just measure the transcription, or even if you just measure the messenger RNAs, it's not necessarily telling you about how much of the protein there is. And because remember, there's all these steps in between. And so Western blot is one, and mass spec are ways that you can get information about this. So mass spec is basically you're measuring all of the proteins. And so mass spectrometry, basically you can cut up all the different proteins into a bunch of little short peptides, and then send those peptides through this mass charge analyzer thingy-bob. And these fragments are going to get separated based on their mass and charge. And then it can be compared to like a database to see what sequences, what protein those came from. So this can be used if you can find all these shotgun all these sequences and then see how much of various proteins were expressed. But there's biases in this method, so it's not. You have to do other things if you want things to be really quantitative, but you can get a relative idea about protein expression under different conditions and that sort of thing. If you want to look for specific proteins, what you typically do is a Western blot. And so once again, we're using antibodies here, antibodies against a protein of interest. And with Northern blot, we saw that we were separating RNA and then transferring it to a membrane. Here we're separating proteins and then transferring them to a membrane, and then we're blotting the membrane using antibodies against the protein. And then we can have labeled antibodies, and then it's going to allow us to visualize how much of that specific protein's there. When we're doing experiments in the lab with like recombinant protein expression, so then when we're talking about expression, we can actually control things a lot better, especially when we're working with bacteria. So we can use like an inducible system where we can have we can have our gene under this promoter, this T7 RNA polymerase promoter. So it's different than the RNA polymerase that the cells use, that the bacteria uses for their own stuff. And so if you can get them to make this T7 polymerase, then you can get your gene to be made expressed even when the bacteria stuff isn't. And we can induce the expression of the T7 polymerase using this, taking advantage of the bacterial lock operand system. And so I have a post on this, but this cool system where basically we add this chemical called IPTG, and then we can get T7 polymerase made and then get our protein made, or at least hopefully. So now when we're talking about like, is our gene expressed? We can see like, okay, if we take a pre-induction sample and a post-induction sample, so this is an SDS page gel. And so we're separating proteins by size and then staining it. So this is before purification or anything, obviously, but you can see that you have this band here that shows up in the post-induction samples or after the IPTG, but not before. And so, yay, our protein was expressed. But of course, If we don't see a band, then things can be a little tricky because maybe it was expressed, but it didn't make it all the way to a final protein. Maybe it was expressed and it did make it to a final protein, but it was insoluble and it ended up in these lumps called inclusion bodies. And so it came out in this membrane fragment instead of in the soluble stuff. And so there's when we're talking about expression here, it's like, well, was it really expressed and then it was degraded? Was it not expressed at all? So there are various things that we can also do in order to see like, okay, well, was our protein made? So one way is that if we were to do a Western blot, or if you might be able to see if the band is a smaller size than you thought it was. So you might see a band, but it's a smaller size, like indicating degradation. Or if you were to purify it out, but then you see a band and then a bunch of bands below it, that could indicate pure degradation. If you have, so especially like if you have a tag on the N terminus, and then you can see if it's like, is it starting to get made? And then it's like degrading or that sort of thing. If you have like a truncation product, or is it getting degraded after the fact? When it comes to cells, I haven't mentioned that you can also, because you're having protein degradation after the fact, one way that you can see if your protein is getting degraded, like, or how fast it's getting degraded, you can actually like use proteasomal inhibitors. So proteasomes, that complexes that is gonna chew up the protein. So if you measure the amount of the protein before and after, like with and without proteasome inhibitors in the cells, you can see if it's getting chewed up by the proteasome. You can also, there are antibodies that you can use that are like specific for ubiquitin. So instead of probing for your protein, you're actually probing for ubiquitin. So you can do like use both and that sort of thing to see if your protein's being ubiquitinated, which is indication that it's getting targeted for degradation. So there are different things that you can do to measure all of these different steps of the gene expression pathway. And often what experiments people are doing is depending on what step in the pathway they're really interested in. As someone who is typically focused on the proteins, it often is like, well, when people are just measuring transcription, I'm always thinking about like, well, what about all those other levels of regulation in between? Is there really a direct relationship? Do they show that the protein levels are actually changing or not changing or that sort of thing? So there's a lot of nuances, as is always in the case of biology and biochemistry and all of this cool stuff, that there's so much going on that it can be hard to get a full picture. But it's important, if we have these pictures at the different steps, then we can see where things are acting